Welcome everyone to the next iteration of the IITE seminar series. Before we begin, I will make one short announcement of shameless advertisement, and that is in the chat. I'm going to paste in a link to a video series. IITE was formed by people like Axel, Rossberg, who's going to be talking today, and one of the initial purposes was educational. So this is actually a lecture series that I gave at Linköping University to undergraduates on using R for data analysis and visualization. So if you and or your students would like to get into R and learn it, here's one resource, at least among the many out there. So you can feel free to take a look. I'm happy to take any feedback on that as well. I'm not going to belabor that. Check it out if you want to. Today, we have Axel Rossberg himself giving a talk. There we go, thanks. And, Axel, as I mentioned, is one of the people behind IITE to begin with. He's at Queen Mary University in London, and he's written a very important and influential book on food webs. And in general, he's interested in large communities. And today he's going to be talking about structural instability in ecological systems. So without further ado, Axel, the floor is yours.
Thank you very much, Guri. And I believe that screen sharing works and you can hear me, right?
Correct, yes.
Okay, very good, brilliant. So yes, this presentation is about ecological structural instability, but importantly about the empirical evidence that this phenomenon exists. And I will number the evidence with these numbers here in the title, and so I will not always tell you if there's new evidence. There's lots of it, and the first, there's evidence for the validity of the model that we use, and among this is this experiment by Gorse, which shows that this logistic equation or the normal form of the transcritical bifurcation applies to single species population dynamics. And another piece of evidence is that if you have two species in a petri dish, you can describe their population dynamics by a Lotka-Volterra competition model. And you have all seen that before. And Gorse finds, yes, all curves are drawn according to equations and match the observations. And if it works for one species and two species, why not that should work for many species as well? So we can just study this Lotka-Volterra competition model with many species. We just have to find out how to choose the interaction coefficients, and we choose them at random. We can choose the diagonal elements as one through normalization, and the off-diagonal elements we choose with independently, identically distributed with some mean and variance. And there is evidence, empirical evidence, that this is the right way of doing it from this paper of Barbier and friends, where they show there is a fingerprint in empirical data of the randomness of the interaction matrix. And so if we are confident in the model, then since the interaction coefficients are random numbers, the biomasses that we get are also random numbers, and we can ask what is the distribution of the biomasses that we get. Fixed random numbers give you fixed biomass in equilibrium. And so we ask what is the equilibrium distribution of the biomass in this model? And for now, assuming that all species coexist. And the equilibrium condition, is simply that this expression here is 0. And then we can solve it line by line. So we start with line number 1 of the j equal 1 and get an expression for the variance of the biomass of the first species. And for some reason, that is complicated to explain, there is this factor 1 minus mu squared here. But you can imagine that if you pull out the expression for species 1 from the sum, then the variance of biomass 1 is somehow controlled by what is left from the sum, and you get an expression that says this: it's the variation in the interaction coefficients times the expectation value of the other biomasses, and the variation of the interaction coefficients times the variation in the other biomasses. And first you see there's a problem, because now you can calculate B1, but you need all the other Bs and all the other variances. And well, how do you do this? We know that we set up the system so that all the biomasses, all the species are statistically equivalent. And so if we know the variance of the first species, we know the variance of all species, we can plug this in. And that's what physicists call a self-consistency condition. And the logic is as simple as that. And once you have done it, then you can solve this system once you know also calculated the expectation of the biomass, which is not difficult. You can solve it for the variance. You get an expression for the variance, and you get an expression from the coefficient of variation of the variance, which looks like this. And if you now test this theory with simulated data, with simulated random interaction matrices, it works excellent. This is the line is the prediction. The dots are the numerical experiments. And what you find is there is this singularity occurring when the species richness becomes too large and the denominator becomes 0 and then negative. You see it here. And the singularity comes from a feedback loop of amplification of indirect interactions. So what is actually happening here is that this species is affected by these complicated interaction terms, but also by variance in the abundance of other species. And this again adds to variance and also of the other species, and again. So through complicated feedback loops with many interacting species, you get this singularity, this blow up of the spread of biomasses, and it comes along with a high unpredictability of what's actually going to happen in the system. Yes, so we get, and this is what we call ecological structural instability. Everything becomes very wobbly, very unpredictable, and it is a result of the amplification of indirect interaction. And the singularity occurs here at this value 1 minus mu squared divided by sigma squared. We come back to that. Now this line of thinking is not new. People have done similar things, physicists, since 1989, and have done it again and again in various different forms, slightly different models. And here, I think at this time, we got a strict proof of the result. Then other people derived it using different methods. What is maybe special from the time that I did it in the book, which Guri mentioned, is that I used an assembly model. I applied the similar method to an assembly model. And maybe more important, I argued in the book, this is not just some interesting play with models, with formulas. This is something that happens in reality. This is so important for understanding what's happening out there. And since then, more and more evidence of this has accumulated, and I want to share this with you today. So yes, let's look at the assembly models. It's exactly the same model, same Lotka-Volterra competition model, but now we add species to it one by one, simulate until we reach equilibrium, and remove those species that go extinct. And you will see that if you do this, species richness in the model increases and then starts to fluctuate around some steady state value. Independent of this, biomass also saturates. And if you now consider a test species that you never invade, you just ask what is its invasion fitness for a given community, this invasion fitness fluctuates. And the distribution over many of these species, kind of, you can draw like this, as of course a standard deviation and a mean somehow. And it controls the probability that a species can invade. So the probability that a species can invade is the proportion of this distribution that is larger than 0. These are all the species with positive invasion fitness. And only recently, we could finally solve this model analytically. So in this book, there is also an analytic solution, but it's wrong because I made some dirty assumptions. Now we clean this up, and it's a long calculation. I don't want to bore you with that, but the result is this. So we have this mu for the mean interspecific interactions, this sigma squared for the variance of the interspecific interactions. And as before, this is the value of species richness at which the singularity occurs, the full structural ecological structural instability. And then you need two other numbers. There is some quantity y, which I won't explain to you, which has a distribution, and you can look what the mean value of y is, and it's this, and the mean of y squared, it's that. You see there is some interesting math involved here. And in the analytic solution, we find the species richness in the equilibrium, in the steady state of the assembly model, is given by this expression, which means the assembly model never reaches the singularity. It stays, the species richness stays somewhat below. And the reason is that at this point already, the system is so wobbly and so sensitive that each invasion generates on average an extinction of a species, and you don't need to push it fully against the singularity to get this effect. We can also calculate the standard deviation of this distribution of invasion fitness, which is this width here, and we can calculate the relaxation time of this random walk of the invasion fitness of test species. And of course the mean, and so the invasion probability, and the relaxation time, or the relaxation, yes, the correlation time is given by this expression here, and the invasion probability, and this is an amazing result, is exactly 1/2. So half of random species that we try to invade into the system can invade under quite general conditions. And here is the kind of just briefly numerical test of the model. The dashed line are the analytic results. The colorful points are simulation results. The limit of large and complex communities corresponds to extrapolating these points to 0 on this axis. And you see it's a very nice fit. And so now we can ask, is this borne out in reality? Do we see these things really happening? And the easiest thing to test is the question about the invasion probability. Do we really see this invasion probability 1/2 in reality? And you see it, for example, here in this paper about the invasion of non-native bird species into Hawaii, and the invasion probability is pretty much 1/2. In this paper here by Jaschke and Stryer, they find using a range of species and averaging over the entire data set that the mean invasion success is 1/2 for introductions from Europe to North America, and 1/2 for the opposite direction. And that's quite astonishing, I think, that such a model, simple model, can explain these numbers. And you can find something similar also for plants. And so I tell these invasion ecologists, if you really want to understand invasion ecology, you have to go through these modeling exercises, and you have to understand the math, because otherwise you're missing something essential. And of course, that's a bit of a hard sell. We say, okay, these indirect interactions are strong. We can quantify that by playing a little bit with the formulas I showed you. One can argue that the impact of perturbations is amplified by the indirect interactions by this factor here. And so that means the 1 here corresponds to the direct effect, and the logarithm of 4 to the indirect interactions. So the indirect interactions contribute 54% according to this theory, which we just saw. And they're independently of this, a colleague called Menge, he investigated the strength of indirect interactions in rocky intertidal communities, and he found that indirect interactions contribute around 40%. And I think that's an excellent match, because there is no other explanation for this 40% number. The method that he used is a bit different from ours in the way that's quantified. And considering this difference, it's an astonishing match between theory and observation. And since you have all these indirect interactions, and they complicate things, and the whole thing is wobbly and hard and sensitive, it's a recurring observation in ecology that in these complicated community models that people build, it's very difficult to make reliable predictions. So Peter Yutzis, he speaks of highly indeterminate behavior, or of enormous differences in the predicted outcomes depending on details of how exactly you set up your model. Or then later, Mark Novak, he says this: indeterminacy rapidly overwhelms predictive insights. In other words, these complicated community models, they're rubbish in predicting what's actually going to happen. And I think we have, as theorists, we have to embrace this. We have to say, yes, this is true. And because that's one of the reasons why so many people are skeptical about theoretical ecology, that whenever we try to build these models, they don't work. We have to say, yes, you're right, they don't work, and we know why. There is a theory explaining that, but there are so many other things that still we can explain and understand, and let's focus on these instead. Otherwise, we will be continuously kind of, I think, have this debate of if our science is of any use or not. We can also, from this same kind of model, argue that invasions are roughly by a factor square root of s faster than extirpations, which then explains why the empiricists find this phenomenon of invasion credit and extinction debt, where they say the extinctions tend to happen much slower than invasion. That's exactly what this kind of model predicts. And some empiricists say the ecology, the theoretical ecology doesn't make sense because we always talk about equilibria, and I never see equilibria in my data. And to some extent, they are right, and that's what they should expect, because if you have a system which is near the limit of ecological structural instability, the relaxation to equilibrium can take a very long time. This one is the intrinsic time scale for a single population. And here you have 100 species or 170 species, and it takes about 170 times longer for the system to relax to equilibrium. And again, so this observation here is understood to some extent, and there is some more theorizing to be done on this problem, but it's to some extent understood based on the theory of ecological structural instability. You can also take a slightly different approach. In the assembly model, we assume in principle there is an infinite species pool. The species we add one after the other is always a new species. But you can ask, okay, if I have a finite species pool, what happens then? And that's the problem that was investigated by Guy Bunin in 2017, and he has kind of made a big school of thought out of that. And what he found is there is this phase diagram where if you have weak interactions and low species richness, you're in this unique fixed point phase. And if species richness and interaction strength increase, you move more towards this multiple attractive phase where the system can switch between different community structures on a heteroclinic network or heteroclinic orbit. And this line here, this black line here, is exactly the point where this communities reach the limit of ecological structural instability, and therefore the community composition is not well defined anymore. Some species are excluded and others come in. And yes, and then they showed recently that this phase transition occurs with increasing species richness or increasing interaction strength. You move eventually from a situation where the populations in a microcosm are stable and don't fluctuate to a situation where they fluctuate widely because of these phenomena, adding to the empirical support for all this. We can also look at food webs. And in order to look at food webs, we have to look at this problem of ecological structural instability in a slightly different way. Instead of solving this equation line by line, we can look at it as a linear algebra problem and say, okay, we have to invert this matrix to calculate the biomasses. And it's well known that if a matrix has eigenvalues close to 0, then its inverse is ill-conditioned. If the matrix is ill-conditioned, there is a high risk or a high possibility that the inverse of this matrix has very large eigenvalues. Everything becomes messy. The whole solution becomes sensitive to small changes in the matrix entries. And as a result, you're unlikely to find only positive biomasses if you have a situation like this, where the spectrum of the original interaction matrix overlaps 0. Therefore, you wouldn't expect this to happen. You would expect the spectrum not to overlap 0. And indeed, if you simulate our assembly model, you find that the spectrum of the interaction matrix starts as a small circle and then it grows, and it then stops to grow as the species richness saturates, and there is a gap. There's a gap remaining, and according to our latest theory, this gap is around 0.24. And so yes, the spectrum never reaches 0. And which also shows that this is not the same as the theory of Robert May from the 1970s, because the theory would predict that somehow you reach the instability and then interesting things happen. This here happens before the linear instability occurs, and therefore linear instability of these systems is not so important as people believe. Okay, but this is just a different way of looking at the same phenomenon. You can look at it in terms of these eigenvalues of matrices, or in terms of these self-consistency conditions, and deep inside the mathematics is essentially the same. The advantage of using the spectra is you can now look at interaction matrices that have a different structure from those of entirely random entries, and do a similar kind of analysis. For example, in complex food webs, you can calculate an effective competitive overlap matrix using this formula. If once you've calculated the competition matrix by solving this matrix equation here, and this overlap matrix has a structure like this. Again, ones on the diagonal, and then some more or less symmetric entries left and right of it. And we could look at the case where this is perfectly symmetric, and we ask, so how does the spectrum of this matrix depend on food web structure? And we find it depends on the ratio of species richness from one trophic level to the next. And if you have a very flat food web, like here, where the species richness declines very quickly, it's flat trophic pyramid, then you don't have so many eigenvalues close to 0. While if you have a steep trophic pyramid with lots of species at higher trophic levels, then you have lots of eigenvalues close to 0, and there's a transition somewhere at the richness ratio of 0.3, where you start getting all these eigenvalues close to 0, implying that you get ecological structural instability, and you don't expect these species at higher trophic levels to coexist anymore. So from these considerations, you expect species richness shouldn't exceed, the richness ratio from one trophic level to the next shouldn't exceed 1/3. And indeed, for example, here people find that the mean ratio of the number of predators to prey species is 1/3 in marine benthic communities. And here in freshwater communities, Jeffries and Lawton, they find the same. The richness of the number of prey species to predator species is the ratio is 1/3. And yes, and here what they call a predator is a species that directly eats the prey. So it's in just the next trophic level, then you get 1/3. In this data set here by Owen Petchy and friends, you see that the species richness declines approximately by 1/3 with each trophic level. If you add species richness, not only which you consider not only the species richness from one trophic level to the next, but you add all the predators that are above a given level, so like say all above the herbivore level, then you have to add these numbers, and you get a ratio of 1/2. And indeed, this is what Warren and Augustine found, a ratio of 1/2, again, for aquatic communities, not only in local communities, but also if you look at all the species around Sheffield or at all the species in the UK. And I think that's quite astonishing, and is evidence for ecological structural instability occurring not only at the local scale, but also at a regional scale in very large ones. And then you can look at entire marine food webs here, for example, in the Barents Sea, where we plotted the body sizes of species on the horizontal axis and the number of species smaller than the threshold body size on the vertical axis, both on logarithmic scales. We find this power law relationship, and we can explain that by saying, yes, we have a decline of species richness by a factor 3 as we go from one trophic level to the other, and this corresponds to some predator-prey mass ratio on the logarithmic body size axis. So this is the evidence also for this ecological structural instability happening in these large food webs. They explain this power law species size distribution, and we can reproduce the same in models, and we can kind of show that this is the underlying mathematics. And then you can look at spatial biodiversity patterns, and we can first look at some simulations of that. Here you have a system of several Lotka-Volterra competition models. Each of these patches here corresponds to one Lotka-Volterra competition model, and they are linked through dispersal on some kind of more or less complicated network. And if you add more and more species, you find that some species are more occupying this part of the network, and other species are occupying more this part of the network. And you can ask, say, what are the macroecological patterns that we get in this system? And let's look first at the gamma diversity, which is this line here, and the alpha diversity, local diversity, which is this line here. And first thing you observe is if you disturb gamma diversity, alpha diversity is pretty much robust, which is can be understood based on ecological structural instability. But even the gamma diversity kind of is pretty robust and goes back to some equilibrium value, no matter in which direction you perturb it. And we can show that this is the result of ecological structural instability on both scales. We can look at first at the local scale. If we reduce the competition matrix, or the effective competitive overlap matrix of the entire meta community to the set of species that are present in a given patch, then we move from this gray thing, which corresponds to the entire meta community, to the black dots. And depending on how you define your local community exactly, you get slightly different matrices and spectra. But overall, what you find is that indeed the spectrum touches zero, as we expect from the zero of ecological structural instability at the local scale. And at the global scale, you can calculate an effective competitive overlap matrix at the by doing perturbation experiments for the meta community. You get and then you get a matrix which has a spectrum that looks like this, which is very similar to the simple Lotka-Volterra model, which I showed you before. Again, it's approaching, the spectrum is approaching the zero, but not covering it. This is a signature of ecological structural instability now occurring at the meta community scale. And you can use this to calculate or predict species richness at the meta community level by looking at the entries of this meta community level interaction matrix. Then there are other patterns that the model reproduces. As you increase species richness and gamma diversity, and finally you get a saturation of gamma diversity, you move from this kind of situation to this kind of situation. And you get, when you reach the limit, the diversity limit imposed by ecological structural instability, you suddenly see the patterns seen in nature, which is a very skewed range size distribution, which is seen in nature. And the other is that the collapse of correlation between abundance of species. So the knowledge that one species is at a patch doesn't help you predicting if any other species is at the patch. And you see this only in the saturated case, but not in these other cases. So that's what people observe in nature. That's what people observe in nature, and that corresponds to ecological structural instability at the meta community scale. You can also look at species error relationships, and they, in this model, they look pretty much what people observe with kind of reasonable exponents. And Brian McGill has said that this pattern here is the consequence of these patterns, and so we won't kind of stretch it as another piece of independent evidence, but it looks good. We can look at spatiotemporal biodiversity. So if you take the same model as before, but make it larger and add more and more species to it, what you find is that initially, as gamma diversity increases, while you add more species, and alpha diversity increases too, but then alpha diversity saturates. And it saturates at the limits of ecological structural instability, as we have seen. And you can still add more and more species, increasing gamma diversity. And at the moment where alpha diversity saturates, interestingly, you start to see the system starts to wobble around, oscillate, and eventually display a continuous turnover of species composition at each patch. So each patch, the equilibrium disappears, and instead you cycle through different species compositions at each patch. So you have a non-zero temporal beta diversity, continuous turnover. And we understand pretty well mathematically why this transition here occurs at the same transition as this transition, which implies that if we see this continuous turnover at the local scale, this is additional evidence for ecological structural instability. And do we see it? Yes, we see it a lot. That was a paper by Denotes et al., published 2014. People are very excited that they see all over the world this continuous turnover of species composition is exactly what our theory predicts. We can do other tests. I'm starting from the right here. This is again the exponent of the species error relationship, but now measured over different time spans for species composition measured over different time spans. And what you see in this empirical data is that the exponent declines linearly with the logarithmic time span. And that's what we find exactly in our model. And you can also look at the species time relationship and ask how does the exponent for that decline with the area, and it declines again logarithmic with the area considered, and that's what we find in our model. You find in empirical data these fluctuations of species richness around some constant value, which we find in our model. Or you can look at the temporal occupancy distribution of species in a patch. So there are, for every patch, there are some species which are always there. These are these here. Some species which just occur once and then disappear. They're very rarely there. And some species are there about half of the time. And this is empirical data. And we find the same kind of structure in this model at the limit of ecological structural instability. And I challenge you to reproduce this with any other meta theory of meta-community ecology. Another piece of evidence developed totally independent from this line of work. It's the observation of so-called dark diversity. It started in 2011, where people said, oh, we find that there are species that should be in a certain habitat, but are not. For example, here the species E, which is also a grassland species, and the species D, which is also a grassland species. And we expect this based on the ecological structural instability, because if there are too many species here, this whole thing becomes unstable and some species go extinct. And so this is an independent, the observation of this dark diversity, the absence of species that should be there, is, I think, additional evidence for this phenomenon. And then people could say, oh yeah, you observed this permanent turnover of species composition. We observed this in all over the world. But isn't this the effect of environmental change? Isn't it so that we see this turnover simply because the environment changes and therefore species composition changes? There's nothing to do with ecological structural instability. And yeah, that may be true. So we tested that. Is it really so? And we argued that if environmental change drives the turnover, then we would expect that the turnover becomes faster and faster, because environmental change becomes faster. At least it started to become faster around the 1980s or 1990s or so. And so we analyzed the same data that the Denotes et al. Analyzed, and asked if I cut the time series and ask how much does the turnover before the cut differ from the turnover after the cut, we find that actually turnover becomes slower. So for example, for this cut here, we see a decline in the median turnover. Decline in the median turnover rate from before the time cut and after. And it is not what you expect if the whole turnover is driven by environmental change. And we find it independently for birds. We find it for these so-called mixed communities. We find it for the benthic communities, and these are the ones that contribute most of the data. And then we also find it for, we don't find it for fish, because fish, of course, are managed and the whole population dynamics is different. So this result, yes, speaks very much against environmental drivers of turnover and for intrinsic turnover. And why does it slow down? Well, we can build a model that reproduces this phenomenon. It's again your Lotka-Volterra competition model with the competition here, and this is all the linear growth rate, and we can simulate a degradation of the environment by subtracting some constant from the linear growth rate. So everything grows slower. And then we find that if we simulate this model, we get this complicated species turnover, that indeed with increasing degradation, turnover becomes slower. And that is because the more the environment degrades, the fewer species are able to invade a given patch, and therefore the overall number of species that can invade is fewer, and therefore turnover becomes slower. And only if you increase the environmental change, which is represented by this term a lot, then you can get an increase in the turnover rate. And so that's the end of my talk, and I say hooray! There are 20 lines of empirical evidence demonstrating ecological structural instability across the real world. And I think, and this is the main point of my talk, we are far beyond the point where we say that ecological structural instability is an interesting idea or some fun thing that comes out of models. I think that at some point you have to say, no, there is so much evidence for it. Of course it is real. Of course we can see it. And we should do this step and say, yes, okay, this is a dominating, very strong phenomenon happening all across ecosystems. And as theorists, I think we should embrace that and say that's fantastic. That allows us to understand and analyze ecological systems in a new way, and hopefully make some progress and show that what our art is useful. I think we have to move away from always looking at systems with 2 or 3 or 4 species, because they hide the truly interesting things that are happening in the real world. And so I hope that with this presentation, I have won a few fellow travelers and joining me on the journey of exploring the implications of ecological structural instability and showing its usefulness to the rest of the world. Thank you very much.
Oh, thank you, Axel.
Oh, they have lots of hands raised now.
I think they're claps.
Okay, sorry.
But hopefully there'll be some hands raising soon enough. If yes, there we go. Well, Axel, do you want to field your own questions, or
Yeah, no, you moderate, and I answer. Yep.
Okay, cool. So Washington, do you want to go ahead?
Sure. Thank you very much. That was a really inspiring talk. I was unfamiliar with many of your 20 lines. I'm relatively new to this field, and I'm fascinated and want to dig in much deeper, and I'll look at your book and some of these things. I have a very specific question about one of the lines that you mentioned, which is this factor of 3, which I found very interesting, and this curious fact that, of course, the sum of the powers of 3 adds up to 1/2. It's beautiful. If you take your models, which produce that fraction, and you artificially have some external agent truncates the chain at the high levels at the high trophic levels, human agents come in and get rid of the top level predators. Do you have any comments on how that changes the species mix? Does that increase or decrease the number of prey species if you get rid of the second level predators, for instance? Any comments on that?
I had a slide on that which I deleted. So what you will see, the slide would show you in a model the same power law decline of species richness with body size that I showed for the Barents Sea. And then I would say, okay, now I truncate the higher trophic levels, and you will see nothing happens to the lower trophic levels. And then I'd say I throw away half of the primary producers. I extinguish half of primary producers. The whole thing simply moves down a little bit. The slope stays exactly the same.
Huh.
Which
So it's still a
demonstrates
factor of a
it's
third,
totally
even if you have an artificial upper bound.
Pardon?
Even if you have an artificial upper bound, you still get the factor of a third all the way up to that bound.
Yes. If I would throw away all the larger species, that would have very little effect on the lower trophic levels.
Fascinating stuff. Thank you very much. Great talk.
Giza?
Also, thank you for the very interesting lecture, and I don't understand most of it, but my basic issue with all that kind of modeling, that in the first type of modeling, when with the completely random interaction, there is a intraspecific and interspecific competition is considered as a completely different phenomenon, which I doubt it is realistic, because it is essentially say that the species can live together beautifully if they don't interact, and the interaction is just a disturbance of their beautiful diversity, which is completely different from the real world. And of course, the trophic level model is completely different, because they depend on each other in a significant way. So I like this much better. And the question is whether do you agree that these things are very different, or you feel that it is essentially the same issue?
I think they're essentially the same. So if you have the predator-prey model and you calculate your overlap model matrix, competitive overlap matrix from shared prey, essentially, then you can have a dominating diagonal and rather small off-diagonal entries if they don't overlap much in the prey they eat, and then you have exactly the same thing. And conversely, of course the intraspecific competition and the interspecific competition is essentially the same thing, but intraspecific competition is usually, I would expect, stronger than the interspecific ones. Within the species, because they all do the same thing, they all compete for exactly the same thing, and so far the competition is stronger. And the bottom line is, there is this paper by Barbier and colleagues who tested that. They did test that. They calculated all the interaction coefficients from experimental data, and they showed it is as this model assumes. If you stretch it a lot and say, in order to make like 1,000 species coexist in the model, you need rather weak interspecific interactions. And then one would say, okay, maybe you're stretching it a lot, but you get these weak interactions once you include space. And you say, okay, some species live more here and others live more here, and therefore they barely see each other, and therefore the interaction is rather weak. And then that's why in order to make so many species coexist in reality, you also need a bit of space. If you try to put everything into a flower pot, you get very few species coexisting.
Yes, so I certainly agree with this picture that the decreasing interspecific competition is the key for coexistence. But what is the point of having a theory which just suppose that we have a very strong intraspecific competition, because we then just assumed
Because that's what we see.
Mhm.
That's what you see. If you actually, as I said, if once you in these experiments where they put plants together and measure their interactions, that's what they see. And it's an unlimited size. And if you would increase the size of these plots and have a bit of heterogeneity, you would very easily get a situation where most of these interactions are weak because different plants are at different places. And then of course you're right. Are we justified in taking the limit of very large species richness? And I don't claim that species richness is infinite. I just say the larger it becomes, the smaller the errors of the theory. And if you look at what point does it start to work reasonably well, the answer is with about 10 to 20 species. So we don't really need so large species richness. That also means you don't really need so weak interspecific interactions as you may think you need. And finally, of course, you get the nice mathematical results in this limit of large species richness, but the principles apply already before the quantitative agreement sets in.
Are there any further questions looking up? I saw Steve, you just turned your camera on, but I didn't know whether that was to ask a question or not. But well, I think one thing I wanted to press you a bit on that last bit is kind of aligning these scales, because I think to some extent you're talking about it just then, but this theory relies on strong, fixed, and random interactions, which kind of we kind of know is not true at really small species levels. And then as we get bigger and bigger scale, maybe at some point it perhaps starts becoming true. And then if you get to even bigger and bigger taxonomic and spatial scales, it starts to become untrue again, because we're kind of losing this kind of cohesive community where anything could interact. I guess the challenge is, how do we make sure those scales are actually aligned? And I think this is the bit particularly where interactions being strong enough to actually be causing the majority of extinctions is a big challenge. Because it's very hard when you look at a species that's gone extinct in the wild, you can't really particularly easily diagnose what is it that's driven it extinct. It often appears that it's some sort of climatic event, whatever, that's come along and just knocked it on the head. Now, was it rare because of the competition that pushed it into that position? Or was it just going about its own business and would have gone extinct anyway without the competition? So I guess the thing is, do we need to be diagnosing a bit better the causes of extinctions to be able to apply these tests that you're looking at?
You're again asking, can I predict somehow, understanding the entire ecosystem, can I predict the fate of individual species? And the answer is no, you can't. We know why you can't, because of all these interactions. So if you ask me why does this one species go extinct, the answer is you can't know. There is no way of knowing with the normal methods. Therefore we use other tests, and I've showed you the tests.
But if we're saying a certain, expect a certain turnover of species,
yes,
I guess these models, pretty much all of that extinction is due to competition. It might be indirect or it might be very direct, but in some ways it's competition driven. And I guess the challenge is, how would we know that a real extinction we've observed, obviously, well, a whole set of extinctions, some fraction of them will be one, some will be another, or various proportions assigned. I feel we kind of need to be getting towards that to be able to.
I think what you can do is you take some species like palm tree, and you plant them in London, and you make sure there are no competitors. Then you will see the palm tree survives. You have a plantation of palm trees somewhere in London, and the palm trees will survive. The only reason they don't spread in London is competition. The other species survive better. And there you have it. That's the answer. So of course, most of these extinctions occur because of competition. But this is not my argument. My argument is that we can make quantitative predictions of what's happening, and these quantitative predictions are quantitatively borne out. And there is no other explanation that anybody has come up with. It's much stronger than that. It's not kind of what do you think or couldn't it be? It's a hard prediction and it's hard verified.
I know we can go on of this for a long time. I just let someone else jump in. Oh, now we've got two. Okay, I think it was a score draw of who put their hand up first. So, Chrissy, go ahead.
Okay, I just wanted to follow up on this palm tree example. Are you arguing that the only reason any species is not found somewhere else is because of competition? Because I can think of several examples that would not adhere to that.
No, I'm not. That's not what I'm arguing.
Okay.
There is obviously environmental filtering, and there is biotic filtering, and people agree on that. And
okay.
The question is, how strong is the effect of environmental filtering, and how strong is that of biotic filtering?
Okay.
And in a nutshell, one of the things we're saying is that this environmental filtering is of course important, but it's estimated. Its importance is overestimated. There is so much going on because of biotic filtering.
Mm-hmm.
And because otherwise these things we see couldn't be understood. Just can't be understood unless you invoke biotic filtering, and then suddenly it falls all into place easily, without effort, without kind of tweaking things somehow. It just happens. And then you can explain what you see. And without biotic filtering, you can't explain what you see.
Okay. Okay. Thanks for clarifying.
Steve?
Very interesting talk. Thank you very much. It's material I was not familiar with, but I need to get more familiar with it. I have first a question about the nature of the theory. To what extent does it depend on assuming that species are coexisting at equilibrium?
In the beginning, that's the first step you have seen that there is an equilibrium assumption, but then it turns out in reality they will not be at equilibrium. That's the final conclusion. What is happening is for each if they have a patch which is isolated with very little bit of very small dispersal into the patch, then you can have one community, and you can have another community, yet another community composed of different species. And as you have this very little dispersal into the patch, every now and then a species invades and it flips and leads to a different community, and then afterwards another different one. And mathematically, that's called a heteroclinic orbit or a heteroclinic network. Okay. And now, if in reality the dispersal is not small, it's large, and therefore kind of you don't see this heteroclinic network and the switching between different states. But in order to conceptually understand what's happening, I think still the heteroclinic network is the best mental model. So that's the dynamics is kind of following somehow along this heteroclinic network, but in a very rough way, because we perturb it quite a lot through invasions.
Okay. But that's sort of the skeleton of it, is this heteroclinic network where it's moving from one equilibrium to another.
Yes.
Yeah. Okay. And there's a lot of evidence against purely random structure. There's evidence There's a lot of evidence for modularity, for example. There's evidence for species clustering. which is, and neither of those is in your model. How much difference do you think that's going to make? Are these predictions robust to these very substantial structural deviations from random interactions?
That's been studied, these questions, and published in a paper in PNAS by also Mathieu Bandbier and Guy Boon and colleagues and Michel Laureau. And the answer is it's very robust. So they said you can even, instead of having a totally random matrix, you can have a block matrix with two guilds which interact strongly, and then between them interaction is weak, and up to some point it doesn't matter, and then you reach some threshold where the distinction between the guilds becomes too strong, and then the theory breaks down. But you can stretch it quite a lot, and the reason intuitively is that the underlying random matrix theory is just as robust as your normal distribution. So this circular distribution you get for the random matrix is a generalization or some other form of the Gaussian distribution of values you get. And both are very robust phenomena. And therefore the whole theory is very robust to structure that you put into these interaction matrices.
Thanks.
Thank you.
Go ahead.
Thank you for the great talk. So I have a follow-up question on the heteroclinic cycles.
Yes.
Yeah, I was very amazed by all the empirical evidence that you gave in the talk, and I wonder if you have any empirical evidence of heteroclinic cycles in mind, because I think that's something that theoretical ecologists talk a lot about, but I don't know, it's just really hard to find empirical results that's supporting this. Potential also because of time scale, and when the community completely change, we don't really know what's going on. So yeah, I want to hear your thoughts on that.
Let me try to share my screen to answer that. Wait a moment. Yeah, here. So this one here, this is empirical data. And you see that there are some species that are there in this local community all the time. Some are always dropping in and dropping out, and others are there only for a short time. And this is kind of the structure that you expect from these heteroclinic cycles.
May I ask why there are species that always exist there? Because for heteroclinic cycle, you would think you would expect everything to sort of only appear for a certain amount of time.
That's because in our simple model we assumed that the intrinsic growth rate, the R's, are all the same. But
okay.
There's some species which with a rather high intrinsic growth rate, they are difficult to kick out of the system, and therefore they always stay there. And
okay, thank you.
The environment, yes.
Thank you so much.
Okay, thank you for the question.
Are there any final questions from anybody? Oh, we have something in the chat. Just seeing if it's a question or not. Can you see it, Axel? And it might be worth reading out the
40
question.
to 50% community alteration seems to be down to the indirect effect as suggested by Menge. But where these are less initially visible, more long-term pressures, the extents of these pressures are less immediately obvious. Exactly the reason why this mathematics is so helpful understanding this progression. So I think that's more like a support of this idea rather than a question, I would say. Yes, and so that's an important thing, that these indirect interactions take a very long time to unfold, and therefore you have these very long transients in these systems, also as a result of indirect interactions.
Okay, fantastic. I think if that's it, we will round this to a close. So thank you, Axel, for
Well, thank you all,
great talk.
thank you all for
And
listening.
well-
Yep. Thank you for the questions.
And yeah, thank you. So we'll have another one in 2 weeks' time, and I hope to see you then. Look out for the email. Thank you.